So you have, after this presentation, I hope a clear idea of what has been achieved and implemented through the first two calls for proposal. And what I will do now is to give you some insight into the project derived from the third call. Thank you. For those who are interested to have, I would say, a general overview of the first two calls, I can recommend to look at this paper, which is freely accessible from the internet, and I think that there are also reprints available outside the room. So the third call for proposal generates a number of projects. I will briefly mention EU AIMS, which is I would say, a unique project worldwide to tackle the challenges of autism, and more precisely of autism spectrum disorder. It's a huge investment, as you can see, a total budget of 36 million. You see the number of partners from pharma companies, academic institutions, SMEs. We are quite proud to have Autism Speak, which is one of the most important charity organizations for autism, which actually is based in US and which decided to invest in this project, which will create a unique clinical investigator network for autism, which will develop, among others, novel imaging biomarkers. There is also an important biobank which is built, which will allow to investigate further some potential genetic predisposing factors, the famous copy number variants. And also, and there will be a link with another project on induced pluripotent stem cells from humans. Indeed, they will generate cell lines from autistic patients, and this will be an important tool to not only investigate the basis, but possibly also to screen for potential drugs. DIRECT is one of the three projects in IMI dealing with diabetes. And in this case, the idea is to develop biomarkers allowing to predict the evolution of diabetes and the responses to drugs. We know that when you make the diagnosis of diabetes, some patients will rapidly become insulin dependent, others not, and it might be very important to predict this and also to better tailor the drugs that you administer to the patient. And there also there will be a large database built, and as you can see, there is already a huge number of patients with type 2 diabetes scheduled to be enrolled. And again, there are clinical trials which should further evaluate those biomarkers, which will be part of this ambitious project with a total budget of more than 40 million euro. VacSafe, you know that vaccines raise a number of questions, not only regarding their efficacy, but also, and very often, you hear more about some allegation regarding vaccination safety, and people tend to forget that they are one of the major achievements of modern medicines in terms of efficacy. So it's very important to develop objective, large-scale methods to assess the safety of vaccines, and this is the goal of this VacSafe consortium, again, gathering major vaccine manufacturers, SMEs, and several academic institutions for a total budget of 30 million euro. Predict TB is an ambitious project on tuberculosis, where preclinical research will be used to test to assess new tuberculosis drug combinations. This is done in close collaboration with the Tuberculosis Alliance, and again there will be their studies where patient data will be analyzed in comparison with the data derived from this preclinical research. You know that liver toxicity is nowadays one of the major concerns when we are addressing the safety of drugs, and especially during the drug development. And you also know that when you pick up potential side effects late in drug development, this is a huge problem to industry because it means that you lose a lot of money, but also for patients because the later you pick up the side effects, the more patients you have which were exposed to toxic drugs. And MIBDLI is developing innovative tests based on, again, on computers in silico models, as well as tests based on induced pluripotent human stem cells. You know that more and more as pharmaceuticals, we use monoclonal antibodies. Just think about the impressive development of monoclonals in cancer and in inflammatory disease. Also, we use recombinant protein. Just think about the interferons used in many indications. We know that those biopharmaceuticals might induce immune responses in the patients, which might neutralize their activity, but which might also induce side effects. So it's very important to understand the basis of this immunogenicity of biopharmaceuticals and to be able to predict their safety. And there, the approach must be completely different of the usual approach with chemical drugs. And this is the objective of this project.